Bacterial physiology. Today, we'll see about what are the essential things for a bacteria to grow, and also more importantly, we'll see about the bacterial growth curve, which is the most important topic for undergraduate studies. So first we have this bacteria, which is mainly made up of water, and it has some basic requirements like our cells, that is your carbon, nitrogen, hydrogen, oxygen, little bit of trace elements, sodium, potassium, magnesium, etc. Same way, bacteria also require the B group of vitamins for their optimal growth. Now once the bacteria start growing, they undergo division by binary fission. So first of all, there is a division of the nucleus where the bacterial DNA unwind and they will form a complementary DNA and two identical molecules are made up. After this phase, the cytoplasm divides via their transverse septum and two bacteria are formed. Sometimes the bacteria stay together with the mother bacteria itself. So that is how the bacterial arrangements are seen. Now the rate of multiplication depends on the bacteria. Some of them, the most common pathogens, are rapid growers, so they multiply very fast. So every cycle happens within 20 minutes. So that means at 20 minutes time, one bacteria divides into two. There are some late ones. The Mycobacterium tuberculosis takes around 20 hours. So your diagnosis, or if you want to grow them in a culture media, it will also take a heavy amount of time. So around 2 to 8 weeks for the bacteria to grow and for us to diagnose the infection. After a certain phase, when the particular dietary requirements are not met and there are toxic products produced because of the cell divisions, slowly the multiplication will stop. Now, to see how the bacteria are growing or how many bacteria do you have, we have two main terminologies. You have what is called as a total count, where we are going to count both the live and dead bacteria. Let us use counting chambers under a microscope and you can view the number of bacteria. But only thing is we cannot differentiate whether they are live bacteria or dead bacteria. The next terminology is a viable count, where we count only the bacteria which are alive. So that is done by a pour plate method. So the solution containing the bacteria is mixed with the media, and obviously the ones which are alive will multiply and grow on the pour plate method. Now we come to the most important topic, the bacterial growth curve. So when you introduce a bacteria into a new medium which has enough amount of nutrients, the bacteria will undergo 4 phases, namely the lag phase, log phase, stationary phase and phase of decline. So what happens in this? First of all, as soon as you introduce a bacteria into the medium, they will have a small period of accumulation of the food products and the enzymes, and the number of bacteria will not increase, but the size of the bacteria will increase. So they are just getting ready for the multiplication phase. This is followed by a log phase, where there is a exponential multiplication, that is the number of bacteria will also multiply. They are smaller in size and they are uniformly stained in your gram staining, and they are very biochemically very much active. This is followed by a stationary phase, where certain bacteria will keep on multiplying and certain others will have reached their end of their life and they will die. So if you count, the total number of bacteria will be more, but there will be a combination of dead and live bacteria which is more or less kept at a particular level. So that becomes like a more gram variable bacteria, and sometimes if there are spore formers, spores will start forming. Along with that, there will be toxin production because of the rapid multiplication of the bacteria. Finally, you have the phase of decline, where the nutrients what you are given are kind of exhausted, and slowly the number of live bacteria will be less and the number of dead bacteria will be more. So this is called as a phase of decline. Apart from this, you have some environmental factors which are required for the bacteria to grow. So we'll see one by one. So first of all, you have the oxygen requirements. So you already know that based on the oxygen requirements, we classify the bacteria as aerobes and anaerobes. So first you have the obligate aerobes. They will grow only in the presence of oxygen, and little amount of carbon dioxide can be harmful to them. Next you have the facultative anaerobes. These are aerobic bacteria which can also grow anaerobically, and some of the pathogenic bacteria which affect us can be classified under this category. Then you have the facultative aerobes, which are basically anaerobes that can tolerate and grow in a oxygen filled atmosphere. Then you have the microaerophilic bacteria, where they can tolerate a little bit of oxygen and they will grow well. Then you have the obligate anaerobes, where they don't want any kind of oxygen, and presence of oxygen can be lethal to them. Finally, you have the aerotolerant anaerobes, which can tolerate oxygen for some time, but they do not use it for their routine multiplications. Carbon dioxide, in a normal bacterial growth, we have a small amount of carbon dioxide added to bacteria. Even in your routine pathogenic bacterial growth, we add little bit of carbon dioxide to enhance certain features like hemolysis. Temperature wise, you can classify them into three types. The psychrophils, which will grow at a very lower temperature; mesophils, which is most of our pathogenic bacteria, which grow at a normal range around 25 to 40 degrees; and the bacteria which grow at a very high temperature are named as thermophils. Now the pH, the normal pathogenic bacteria generally grow between 7.2 to 7.6. Few of them can grow in a acidic temperature, that is acidic pH, that is the lactobacilli, and some of them can grow under alkaline atmospheres, which is, your example will be Vibrio cholerae. Light requirements, the bacteria can grow well in darkness. Some of them are sensitive to ultraviolet rays, so that is one of the methods used for sterilization. Some of them produce a pigment on exposure to light. Osmosis, just like a normal cell, if there is a sudden hypertonic exposure, the cell will shrink, and the hypotonic solutions will make the bacteria swell and rupture out. Moisture, mostly because the bacteria is made up of 80% of water, drying can kill the bacterium, where some of them can tolerate and live in a minimal amount of moisture for a long period. Slow freeze drying is one of the methods which is used for preservation of these organisms. So this is about your bacterial growth curve and the normal physiological requirements of a bacterium. I hope this video is short and easy to understand. Thank you.